All right, well, maybe we'll go ahead and get started. So my name is Lauren Moore. I'm the Chief Scientific Officer and VP of Research here at the National Ataxia Foundation. And I want to welcome you all to our first science showcase of the year. So this is a webinar series where we highlight different grants that we have funded and the great work that's been done by the research who received grant funding. So today I'm very happy to introduce Dr. Jeannette Hübener-Schmid, who's coming from chiming in from Germany today. And she'll be presenting on work that came out of her 2020 NAF Early Career Investigator Award, entitled Development and Validation of a SIMOA-based mutant Ataxin-3 Immunoassay for Biomarker Studies in SCA3. Let me just a little bit about our speaker. Dr. Hübener-Schmid is group leader of the study of biomarkers in neurodegenerative diseases at the Institute of Medical Genetics and Applied Genomics at the University of Tübingen in Germany. She received her PhD in neuroscience from the University of Tübingen in 2008, and she has over 20 years of experience in the field of biomedicine, human genetics, neurogenetics, and she has authored 43 scientific publications. So without further ado, welcome and thank you, Dr. Hübener-Schmid, for joining us today.
Yeah, welcome everybody. I'm happy that I can present today my my topic about an SIMOA-based immunoassay, which we developed for mutant Ataxin-3. And as my internet connection is not the best one, I would prefer to switch off my video for the talk, but I will switch on afterwards for the discussions again. I hope that's fine for you. Thanks for the warm welcome and for the introduction about myself. But I will give you a very short background. I am biologist, studied in Hanover biology. And as already mentioned, I have my own research group, which is working on biomarkers in neurodegenerative diseases, mainly in SCA2 and SCA3. And we are a multidisciplinary team. And the main focus of the working group are molecular biomarkers. I worked with different groups on, for example, neurofilament light chain, but also GFAP. And my main interests are the disease proteins, mainly for polyQ scars, and here mainly for for SCA2 and SCA3, Ataxin-2 and Ataxin-3. And we are interested in different molecular markers or molecular biomarkers, which could be potential markers for disease progression, but also for therapeutic response, for conversion from the pre-ataxic to the ataxic phase, but also as biomarker staging. My project was funded, as mentioned, in 2020 during Corona, and as a Young Investigator SCA research grant. And the title was The Development and Validation of a SIMOA-based mutant Ataxin-3 Immunoassay for Biomarker Studies in Spinocerebellar Ataxia Type 3. We had two most important aims. The first aim was the development of Ataxin-3 SIMOA-based immunoassay to measure specifically the mutant Ataxin-3 protein levels. And the second was the validation of the mutant Ataxin-3 in human biofluids and comparing different biofluids, but also comparing the levels, the protein levels of Ataxin-3 to clinical data, but also have a comparison of already known other Ataxin-3 specific immunoassays. And we performed additionally in this time development of an immunoassay for total Ataxin-3, and total Ataxin-3 means that we will may be able to measure the wild type form, that means the form without the mutation, and the mutant form, that means the mutated polyQ expanded protein version. Why we need ultra-sensitive immunoassays? That's really easy to answer, because we know that the disease proteins, here especially Ataxin-3 as a disease protein in Spinocerebellar Ataxia Type 3, is very low expressed in cerebrospinal fluid, but also plasma and serum. if we here take a look to a screenshot of the GTEx portal in which the RNA expression of specific genes can be recorded or are recorded, then we see that we have really less amount of this RNA available in the whole blood, but also in CSF, cerebrospinal fluid, and peripheral blood components. And if we think about potential therapies, as you all know, protein lowering therapies are now coming to the market and are evaluated. And to measure the protein lowering, and in this case, the disease protein, which get lowered by so-called antisense oligonucleotides, which binds to the mRNA of Ataxin-3 in this case, and lead maybe to a degradation of this RNA, and therefore to not protein production, or maybe lead another way to an exon skipping and to a shortened form of Ataxin-3. And as readout for this lowering therapies, the disease protein itself can maybe be measured, but we need a specific technique to really be able to measure very small amounts of this protein as target engagement, for example, in peripheral blood, but also CSF. Which immunoassays are already known. There is a highly sensitive immunoassay based on the so-called TRFRET technology, which means time resolved fluorescence energy transfer. And this was published 2020 in my group, and we set up two different assays. One assay which was specific or which should measure the total Ataxin-3 in human biofluids. We used the best known antibody for SCA3 or for Ataxin-3, the so-called Ataxin-3 clone 1H9 antibody, and second antibody, which is binding to the N-terminus of the protein. And by this, we were able to measure the protein levels of total, that means of wild type form and mutated form, in peripheral blood cells, here specifically in PBMCs, peripheral nuclear blood cells, but not in serum and plasma. And if we compare the protein levels of Ataxin-3, total and wild type, total including wild type and mutant form, then we see that we cannot discriminate between controls, pre-ataxic and ataxic mutation carriers, which is expected because we are measuring at the same time also the wild type form. And then we set up an additional assay which specifically recognized the mutated version of the Ataxin-3 protein. Therefore, we used again the Ataxin-3 clone 1H9 antibody and a protein which specifically bind to an elongated polyglutamine stretch, the MW1 antibody. And here we see that we have mutated protein available in pre-ataxic and ataxic mutation carriers, and we also recognize that this is correlating with the disease duration in this case. That means we were able to set up total mutant Ataxin-3 assay on this highly sensitive platform, but in the end, we recognize that we're only able to measure in peripheral blood mononuclear cells or in tissues or in cell cultures, but not in cerebrospinal fluid and not in plasma or serum. Therefore, an so-called ultrasensitive immunoassay is needed, and ultrasensitive means that this assay will detect in the femtomolar detection range. There are already two kind of immunoassays known. One is a mesoscale discovery Ataxin-3 mutant assay, which was published 2020 by Prudencio, and demonstrated that Ataxin-3 can be measured, or mutant Ataxin-3 can be measured in serum and plasma. And 1 year later, the same group demonstrated that's also possible to measure Ataxin-3 levels in urine. We, together with Evotec, a German company, and the European Consortia for SCAR3, so-called ESME, developed different immunoassay also on an ultra-sensitive platform. It's a so-called SMC Pro Ataxin-3 immunoassay. I will go a bit more in detail and explain the differences of the different assays, but importantly, both ultra-sensitive immunoassays were able to measure mutant Ataxin-3 levels in cerebrospinal fluid and peripheral blood, like serum and plasma. Here you see a cartoon of the ultra-sensitive immunoassay technology SMC Pro. SMC is the abbreviation for single molecule counting, and this is a microparticle-based technology, which you see here. And on this microparticles, we have a capture antibody bind. This capture antibody is able to to conjugate the target of interest. And if this is done, then you can incubate with a detection antibody, as a secondary antibody. And this detection antibody have fluorescence tag. And if this binding occurs, then you can have evaluation afterwards, and you're measuring in this 384 plate system by using a robust digital SMC module, which counts photons. And you have here this small interrogation space illuminated by a laser. And if a single fluorescence labeled molecule is crossing this interrogation space, then it generates intense flashes of lights. And in the end, the total sum of all photons are recorded. For the SMC Pro immunoassay setup, we use the same antibodies as which we already know from the highly sensitive TR-FRET assay. We use the clone Ataxin-3 clone 1H9 antibody and the NW1 antibody, which binds to elongated polyQ stretches. And we set up first using recombinant proteins the detection range, and you see that the polyQ expanded Ataxin-3 is measured more sensitive compared to the non-elongated or not to the wild type form. And in the end, we were able to describe the assay performance by the lower limit of detection, which is 0.2 picogram per milliliter, and the upper limit of detection was 427 picogram per milliliter. Then we measured different biofluids, including plasma, CSF, and serum, which get diluted first, and then the assay was performed, and afterwards we assessed the different levels. In our validation cohort, we were able to discriminate to 100% between controls and pre-ataxic, but also controls and ataxic mutation carriers in plasma, Ataxin-3, but also in serum. And importantly, we received the similar results in serum and plasma. Afterwards, we compared the mutant Ataxin-3 levels cross-sectional to the clinical data of our patients or of our mutation carriers, which we recruited in the ESME project. ESME is the European Spinocerebellar Ataxia Joseph Disease Initiative, which is recruiting patients or mutation carriers in Germany, in Netherlands, UK, and in Portugal. And currently we have more than 400 mutation carriers collected and standardized, phenotyped, and we used the clinical data which we got and correlated the plasma mutant Ataxin-3 levels with age at onset. We saw a negative linear correlation. We saw no correlation with the INAS, with a non-ataxic INAS score, and we saw or found a positive correlation with the SARA score. That means correlation with age at onset and SARA could be described. In the last months, we analyzed our cohort, our 3 SME cohort, longitudinally, and analyzed the Ataxin-3 levels measured by the SMC-Pro immunoassay in the longitudinal fashion. And normally, if the parameter which we are analyzing is a progression marker, then we see a curve like here demonstrated in SARA, which is increasing over time. But if you check for Ataxin-3, then you see that Ataxin-3 is quite stable over the time. This we also described earlier in the cross-sectional fashion. And now with the longitudinal data, we see clearly that Ataxin-3 is not a progression marker, but maybe a target engagement marker. As target engagement marker, it would be important to also measure good Ataxin-3 or to measure Ataxin-3 levels also in cerebrospinal fluid, because you know that currently the antisense oligonucleotides are injected to the intrathecal in an intrathecal manner, and therefore the CSF is the primary site to measure Ataxin-3 levels, to decide if the substance could lower the Ataxin-3 protein levels or not. And therefore, if we take a look to our SMC-Pro assay, then we see that in CSF, we have really low levels of Ataxin-3 available. In our case, 5 picogram per milliliter, which is quite low and lower compared to plasma Ataxin-3 levels. And if we compare this to the study from Prudencio with a mesoscale assay, then you see that here for SCA3, the levels are lower than what we are measuring with our SMC-Pro assay. We are here in a range of 0.2 picogram per microliter. That means, nevertheless, in which which assay you are using, Ataxin-3 is near background noise in CSF, and therefore maybe we have the problem that we cannot measure and protein lowering of the protein under therapy using these assays, because the levels of Ataxin-3 are too low in this compartment. And therefore, we ask the question if maybe the change of the platform can lead to an improvement of the measurement, and therefore we set up together with the NAF funding the SIMOA-assay, and the SIMOA-assay is also a so-called ultra-sensitive technology. You have comparable to the SMC-Pro assay paramagnetic beads, which you are labeling with your capture antibody, and we have a secondary or detection antibody, which have a biotin label. And in the end, we have an incubation with beta-galactosidase labeled streptovidin, and this we can measure. That means technically it's similar. We have this capture bead, we have this paramagnetic bead, which is a capture antibody, which we incubate with our biomaterial like CSF, plasma, serum. If an analyte is binding, then we can incubate with a detection antibody, which is biotin-lated. And if this is done, we can load the beads into this SIMOA array, which have a size of 216,000 femtoliter each well, and the well therefore is only large enough that only one bead can include it. If together with the beads or with the labeled beads, we also add the enzyme substrate, which is resorphin beta-D-galactopyranoside, and afterwards this is sealed with oil. And with a laser, we can then immediately or after an incubation time measure the target analyte, because the substrate will be converted to a fluorescent product, so-called resorphin, and this can be here detected in the wells in which you have a complex, and this you see a fluorescence signal, and the other not. Okay, let's come to the first aim of the NAF funded grant. Also for comparison reasons, we used the already known antibodies, Ataxin-3 clone 1H9 and the polyQ specific MW1 antibody. We tested the antibodies both as capture and as detection antibody, but in the end it turns out that the 1H9 antibody was the best as a capture antibody, and the MW1 was best to use as detection antibody. As this all are antibodies commercially available, they are stored in different preservatives. Therefore, we need first to purify the antibody. We tested different agarose, here protein L and protein G, but in our case, protein G worked more perfect, or we found out that protein L was not able to bind the 1H9 antibody at all. Then we had to exchange the buffer from heaters to PBS to avoid interference with the immobilization reaction. And then we tried different concentrations of the 1H9 antibody for immobilization, and this in the end was 0.2 or 0.5 milligram per milliliter. And finally, we used 0.5 milligram per milliliter. The second antibody was the MW1 antibody, which we ordered from Merck Millipore as detection reagent, and this got biotinulated. First, again, we had to remove the tris, which is a buffer substance, to avoid here interference with biotinylation reaction. Then we tested different bioteins to antibody molar ratios. Here 4 different were tested, 5-fold, 10-fold, 20-fold, and 100-fold. And we finally decided for biotin to MW1 ratio of 20:1. With this setup, we measured our exploratory cohort, in which we included 3 controls and 3 mutation carriers. And from each, we had plasma available and serum available. And you see that, as before shown for the SMC Pro assay, we were able to measure in both similar amounts. But what you already also see is that we have a high variance between the different SCAR3 mutation carriers. At this time point, we had to reorder our antibodies and to again label them and with biotin and to the paramagnetic beads. And in this time, we got problems with the MW1 antibody. The supplier did not provide any good antibody lots anymore, and therefore we had a lot of problems in performance. We ordered several lots from the company, but in the end, we had not the possibility to improve the assay or to come back to what we had before as background to noise ratio. And therefore we decided to order a new MW1 antibody from a different supplier. In this case, Study Hybridoma Bank in Iowa, US. And here we had again to optimize our reaction. Therefore, we first had to perform different purification steps because this MW1 antibody was a bioreactor supernatant. Therefore, we had to implement different purification steps to have a perfectly purified antibody. Then we tested 2 different bioteins and also tested different biotin to antibody molar ratios. Again, in this case, 5-fold, 10-fold, 20-fold, and 100-fold. And here for this antibody, we finally worked out with a biotin to MW1 ratio of 10:1. All different steps, as I reported up to now, we are performed on the Quantarix SRX platform, which allows to optimize. And later, after optimization, we use the HDX analyzer, which you see here. And after final establishment, all assays were performed on the automated, this one, HDX analyzer. To further optimize at this step, we performed again different steps to reduce high background noise, which we had initially. We tested 5 different buffer conditions, including commercially available buffers, but also safely prepared ones. We compared 2-step versus 3-step assay protocols. You see here the difference for the 2-step protocol. The capture beads were incubated with sample and calibrators, and afterwards with the detection antibodies. And in the 3-step assay protocol, you have in between a washing step. We tested also different antibody concentrations, 1 microgram per milliliter and 1.4 microgram per milliliter, and also established different streptavidin beta-galactosidase conjugate concentrations, 100 picomole, 150 picomole, and 200 picomole. This is a standard curve of the optimized assay. It's a 7-point standard curve of a recombinant Ataxin-3 with 62 glutamines. And you see that our dynamic range is comparable to the one which we also received in the SMC Pro assay. The lower limit of quantification is 0.2 picogram per microliter, and the upper limit of quantification is around 600 picogram per milliliter. After establishing the assay, we used different biofluids to validate our assay and to compare this to clinical data in our AIM-2. This is again the exploratory cohort, which we performed with the MW1 antibody from Merck Millipore, which shows bad performance after reordering. And in our validation cohort, we found slightly different dynamic ranges and slightly different performance of the assay. But in the end, we could clearly discriminate between controls and pre-ataxic and controls and ataxic mutation carriers in plasma, serum, and also in CSF. The area under the curve, or the possibility to discriminate between healthy controls and mutation carriers, was 100% in CSF and in serum. And for plasma, we had some outliers, but for controls versus pre-ataxic, we had 99%, and plasma and healthy controls versus ataxic, we had 97%. Similar to other assays, we could not discriminate between pre-ataxic and ataxic mutation carriers using this assay. Afterwards, we correlated with the clinical data, which we received from the ASME cohort. And we see again a negative linear correlation with the age at onset. but in this case, we did not found any correlation with the SAVR score or with the INA score. That means here we did not see any correlation to clinical scores. But for the first time, we found a correlation between CSF and plasma, but it's very important to see that we have only 7 mutation carriers from which we received CSF and plasma on the same time. Therefore, a repetition in a larger cohort is absolutely needed here. If we conclude this, then we were able to establish in SIMOA mutant Ataxin-3 immunoassay. We measured mutant Ataxin-3 in plasma, serum, and CSF in a reliable way. We found an area under the curve or a discrimination potency of 1 or 100% in CSF and serum and 99% of plasma in plasma, comparing healthy controls and all mutation carriers. We had a correlation with the age at onset, but not with clinical scores. And for the first time, we were able to see that plasma and CSF may be correlating. This was not shown before by our SMC Pro or the mesoscale assay. If we now compare the different platforms for the CSF measurement, then we have here again the mesoscale assay in which we were able, or in which the group of Prudencio et al. Was able to measure 0.2 picogram per microliter mutant Ataxin-3 in CSF using our Singulex or SMC Pro assay. We were in the range of 5 picogram per milliliter. And in a few cases, which we measured here using the SIMOA assay, we had scaled up to 40 picogram per milliliter. The assays are a little bit different. They are different on the platform, but they are also different on the antibodies used here. The SMC Pro and the SIMOA working with the same antibody combinations, and the MSD is using a different polyQ specific antibody. If we compare the different ultrasensitive techniques, then we see that this SIMOA, that's the SIMOA and the SMC Pro or Singulex assay both use paramagnetic beads, and the Singulex is based on a fluorophore, which is coupled to the secondary to the detection antibody. Here by the SIMOA, we have a substrate streptavidin and beta-galactosidase, which is maybe more specific. It could be discussed. We use different antibodies. The mesoscale used a different polyQ specific antibody compared to the Singulex or the SMC Pro and the SIMOA. All of them discriminate well between mutation carriers and controls. The dynamic range of the SMC Pro and the SIMOA is comparable. For the mesoscale, we don't know, it's not published, but they measured in picogram per microliter, and we measured in picogram per milliliter range. The correlation to clinical data, all of them showed a correlation to the age at onset of the mutant Ataxin-3. And here in the Singulex or SMC Pro, we also saw a correlation to SAVR. In the MSD, they found a correlation to gait, and in the SIMOA, we found additionally for the first time the correlation of CSF and plasma mutant Ataxin-3. And also, it seemed different, the amount of mutant Ataxin-3, which maybe can measure it by the respective technique, but all of them used only a very small group of mutation carriers and controls, and therefore here larger amount are needed. If we compare the different ultrasensitive techniques, then all of them discriminate well between controls and mutation carriers. We have different ranges of sensitivities in all assays. We have the problem for all of them that in CSF we have the lowest levels of mutant Ataxin-3 compared to peripheral blood, for example. In my opinion, a direct comparison of all known techniques is needed. That means that the same sample, sample would be measured in all of these techniques to compare them directly, and all of them need to be evaluated for if they are able to measure under protein lowering therapies, for example, in CSF. As I told you at the beginning, we not only established a mutant Ataxin-3 assay. During the project, we decided also to go for a total Ataxin-3 assay, and total means that we can measure at the same time point wild type and mutant Ataxin-3 protein. For this, we used different antibody combination. We again used the 1H9, which we know from all the other assays, but we also used other known commercially available rabbit monoclonal Ataxin-3 antibody. It's called 13H9L9, and the epitope on the data sheet corresponds to the isoform 2, and we in our lab determined the epitope more closely to the C-terminus of the Ataxin-3 protein. And this assay is really working well in our lab for Western blot, for filter trap, or for aggregate analysis using filter trap or DDH, but the antibody is not working well for immunohistochemistry and immunofluorescence. This antibody combination we tested both as, again, capture and detection antibody, and you see here typical curve during development. We have here the example also during development for 1H9MW1, which is for the mutant version. And you see in gray the combination 1H9, 13H9L9. This is the gray line, and the other way around. That means 13H9L9 as a capture antibody and 1H9 as a detection antibody is this blue line here. And as the gray line looks more similar to the red line, we decided that the 1H9 should be used as capture antibody and the 13H9L9 as detection antibody. But what you can see already is that the curve is quite flat and that we have a high background noise in this assay. And therefore, we started with the optimization and, for example, compared different buffer compositions. You see that this is changing a bit, but not that much. And then we checked for different detergents and different beads on their performance. You see that they are here comparing two different detergent and without detergent and different beads, that the curves are nearly the same. And in the end, with this data, we compared here the non-expanded polyQ, the wild-type version of Ataxin-3, and it's a green line, and the polyQ expanded, the blue line. And you see that we have this strange curve here, which is not explainable. But again, that we have a high background noise and a very flat curve. And therefore, on this stage, we decided to not go for further improvement because all what we did before did not improve. And therefore, we decided not to further optimize this assay. And therefore, currently, there is no total Ataxin-3 assay available in our hand, which can reliably measure total Ataxin-3. In conclusion to the total Ataxin-3 assay, total means always wild type and mutant form of Ataxin-3 because there's no specific antibody available which specifically bind a wild type Ataxin-3. And the 13H9L9 is a very nice antibody, monoclonal antibody, which show good performance in our lab, at least for protein, soluble protein levels, and for aggregated assays, not for immunohistochemistry or immunofluorescence. But so far, we were not able to establish a SIMOA-based immunoassay which show a good performance. With this, I will thanks all my collaboration partners, especially Jennifer Faber, my boss Olaf Ries from the Department or Institute of Medical Genetics, and all this work was done together with the Natural Science History Institute from Reutling, especially Jens Göpfert and Heiko van Beek, which here performed a lot of optimizations to further improve the assays. Thank you for the attention, and I'm happy to take questions.
Great.
I will come back.
Thank you so much, Dr. Hübener-Schmid. We do have a couple of questions. Our first question is, what is the basis for the selectivity of the MW1D2 antibody? How does it distinguish pathologic polyQ from long yet normal polyQ?
This is the MW1 or also other polyQ specific antibodies, which we know specifically bind to polyQ repeats, and there are different publications or different experiments done, which showed that the different antibodies can bind to specific lengths of polyglutamine repeats. And for the MW1, for example, it's shown that it needs at least 15, but could be more often maybe, but also more pronounced to an elongated polyQ stretch.
Thank you. We have one person who says, thanks a lot for this nice overview and congratulations for the great work. Do you know if there is a correlation between brain tissue and CSF, of course, established in rodents or non-human primate models?
We did not use for the ultra-sensitive assay rodent models, also did not compare tissue with CSF, for example. For the highly sensitive assay, we also measured in tissue like brain tissue. And there we found at least a correlation with the aggregation propensity or with the aggregation state. As more aggregates we found, as reduced was the full length or the soluble levels of the mutant version. But we had never checked tissue versus CSF, but it's a good idea. Thanks.
Have you checked if there is a correlation between mutant Ataxin-3 levels and the number of CAG repeats in your assays? If there are results where they may be adjusted to CAG repeats number?
Yes, we did. And the CAG repeat is influencing the binding of the MW1 antibody, this we know. And we also see a correlation. Even this correlation is not quite strong, but we see a correlation between the repeat length and the level of mutant Ataxin-3, which we are measuring with the assays, yes.
Two more questions. Again, if any of the attendees have more questions they'd like to ask, please throw those in the Q&A. But how could SIMOA or other ultrasensitive techniques be scaled up for use in the clinic after validation? What challenges do you anticipate with expanding into the actual clinic with these types of assays?
The assays can really fast scaled up to a large amount of samples, because you can measure a high amount of samples at the same time point. Most of them are working with 384 plates or something like this. That means if you work in duplicates and plus standard and so on, a large amount of samples can be measured at the same time point, and therefore large scale is possible. And for the SIMOA and for the MSD, for the mesoscale, also multiplexing is possible. If you, for example, think about neurofilament, which is an important marker for neurodegeneration, then in this case, if you work on the SIMOA, the NFL is also working on the SIMOA, multiplexing would be possible, that you can measure both proteins at the same time on one platform in one sample.
Do you anticipate any issues, though, with implementing it in, for example, a clinical trial where you have multiple sites?
For the SIMOA, you have this fully automated technique. That means you set up your samples and your buffers and everything, but afterwards everything is pipetted automatically, which is an advantage to the SMC Pro, in which you need to pipette by your own each step, which could be an advantage of the SIMOA assay. But for sure, we have inter and intra plate variability as known for all assays. And the best comparison is always the samples which you have on the same platform or on the same same multi-variable plate or something. But for sure, you have an inter and intra plate variation. And this is for all of them a disadvantage for multicentric studies, that you have a variance if you measure on one laboratory and on the other. But this is for all currently available ELISA technologies that you have this variability.
Is there a standardized protocol for how to deal with biofluid samples and store them prior to assessment? For example, do you add protease inhibitor? Do you not? What specific buffers you use? And how can we deal with that with knowing that a lot of these biorepositories may have different protocols for how they're freezing CSF and freezing plasma?
In the ESME cohort, we have used everything highly standardized. That means all centers are using the same biosampling protocols, but also the same storage protocols and so on, which is leading to less variable results and variances, which is very important. But for sure, if you are using multi-center studies, or if you aliquoting the blood in different centers and so on, there should be difference. Currently, we did not know how good the assay perform on different tube systems, but this is our next step, that we compare, as done for neurofilament light chain, different tube systems, and compare how this is influencing the measurement or the amounts, but this we currently not know, but this is our next step.
All right. Any last minute questions before we close for today's session? Feel free to throw those into the Q&A. But if not, let's all thank Dr. Hübener-Schmid for her wonderful talk today, and I hope you all have a wonderful rest of your afternoon or morning or evening, depending on where you're calling in from. Thanks again.
Thank you so much.
Bye-bye.